this is Todd Herron here for the webinar on CELLvo Matrix Plus, and also to present data on optical mapping and using Cartox voltage sensitive dye system. So in part, hello.
Okay, y'all can go ahead anytime. I just close it because I hear it out here.
Welcome, everyone. Thank you for joining us today. I'm Iris from STEMbiosis, and we are your hosts for today's third session. This session is going to wrap up where we left off from last week. Last week, we talked about cell-derived matrices and for use of them with iPS-derived cardiomyocytes. So today, they're going to go into more on the analysis portion. I'll introduce Travis Bock here from STEMbiosis, and he can introduce the team. Thank you all.
Hi, I'm Travis Bock. I'm the Chief Technology Officer with STEMbiosis. I've been with the company for 5 years directing R&D here, and I've been collaborating with Doctors Todd Herron and Monteiro Da Rocha for the last, I guess it's been 3 years now. They might be able to correct me on that, but it's been a few years now, and really enjoying the work we're doing and very excited about the results that are coming out of this collaboration and the new products that we think can really benefit the field. And so, as Iris mentioned, this is the third part of our seminar series. The first 2 parts discussing selection of cardiomyocytes and selection of substrates for those cells can be found on our website at www.stembiosis.com. And today we're going to get more into the assays. And so with that, I'm going to turn it over to my collaborators, Doctors Herron and Monteiro Da Rocha at the University of Michigan, to discuss options for assaying cardiomyocytes, as well as a discussion of the novel Cartox optical mapping system and Cartox analysis software that has been jointly developed by our groups. Thank you, and I'll hand the floor over to you, Todd.
All right, thank you, Travis. So yeah, I'll be talking about what to do with your cardiomyocytes once they're mature. Before getting into that, I just wanted to present slides just to remind you what the cells are going to look like when you put human cardiomyocytes onto Matrix Plus coated plastic dishes. So here it shows cardiomyocytes, same batch. Cultured on Matrix Plus, the cells become rod shaped and they align relative to one another within the cardiac monolayer. Also, just to remind you, the cardiomyocytes become rod shaped, and this is very robust, where the cells cultured on Matrix Plus become much less circular, much more rod shaped, as you can see in the images of the immunofluorescence. Also, just to remind you, besides electrophysiological and structural maturation, we also see that Matrix Plus promotes maturation of mitochondria, as shown here, with imaging for mitochondria by mitotracker red staining. Also, the increased red staining of the mitochondria indicates that the mitochondria are more healthy and more robust in function than when they're cultured on Matrigel. And then finally, I didn't show this last time, but this is a molecular analysis showing that human cardiomyocytes cultured on Matrix Plus will increase their expression of cardiac troponin I, which is an important and well-known marker of cardiac maturation. And so you can see for the CTNI specific antibody, much more robust immunoreaction at the cardiac troponin I molecular weight. So as the title says for the webinar, now that your cells are mature, what's next? So I've taken this figure from a review article. Actually, it's a best practices article published by the FDA and SIPA committee, which is part of HESI, which is the Health and Environmental Sciences Institute that manages and takes care of validation studies for regulatory science. So there are different options or assays for interrogation of human cardiomyocytes. Panel A shows transmembrane recordings. These are electrical or optical recordings, and they may be intracellular action potentials measured by microelectrode recordings, which are very direct measures, but are very low throughput. And more recently, the field has started to use voltage sensing dyes or voltage sensing proteins, where the cells become fluorescent, and then you can record changes of fluorescence of the fluorescent label. And panel B shows another option for interrogation of the cardiomyocytes, and that is extracellular recordings of field potentials. This is a common approach in multi-electrode arrays, where multiple electrodes are embedded in the cell culture plates and are used as recording electrodes, and also can be used as stimulating electrodes. And in panel C, less direct measurement of electrophysiology is measurement of contractility and motion. And that can be done by measuring calcium transients, which are measured using fluorescent indicators loaded inside the cardiomyocytes. Or there's also approaches using impedance measurements, where you can measure contractility of the cells and record changes induced by drugs that may cause arrhythmias. And finally, for contraction or motion, there's also detection of pixel movement by video analysis. So this table is from the same manuscript for the best practices and use of human cardiomyocytes for in vitro toxicity screening. And here in this table, you can see the technologies for interrogation are listed in the first column. The repolarization effects that can be studied with those technologies are in the next column, and depolarization effects that can be detected in the next column. And then finally, the last column on the far right ranks the electrophysiologic granularity. And so at the top, where there are 3 pluses, the granularity of the assay is high, and at the bottom, where there's only 1 plus, the granularity is much lower. So what we do in our lab and what Cartox focuses on is using voltage sensing optical recordings, where we can detect changes in repolarization by detection of action potential duration 90 prolongation. We also can measure the upstroke velocity, the amplitude, and also the conduction velocity of the electrical impulse. So this is the Cartox machine that's been developed at Cartox and now by STEMBiosis. This instrument was made with funding from the National Institutes of Health, from an SBIR grant from phases 1 and 2. And this machine enables high throughput electrophysiology assays, where you can measure human cardiac action potentials with high spatial and temporal resolution. Also, human cardiac calcium transients can be measured. And we can quantify arrhythmia mechanisms now using mature cardiomyocytes combined with the voltage sensing optical dye detection enabled by the Cartox machine. So this is what the machine looks like. It's made for 1 multi-well plate at a time and can be loaded and is under environmental control. And the kinds of assays or readouts that you get are shown in this figure. So here, this is looking at human atrial cardiomyocytes and measuring the conduction velocity of action potentials through the monolayer. You can see there's a spontaneous rotor that maintains the activation of the monolayer. And when we give a hERG channel blocker, in this case E4031, we can see that there are profound effects on the conduction and development of multiple pacemaker sites, as well as collision of activation waves. And this allows analysis of very complex arrhythmia mechanisms. So as I said before, the Cartox machine also enables high throughput screening of 96 well plates. Actually, any multi-well plate can be used. Here shown is a 96 well plate. These are spontaneously contracting cardiomyocytes. The cells are loaded with Calbright dye to monitor the intracellular calcium flux. And you see there very high efficiency of plating of the monolayers, where all 96 wells contain alive, contracting, and electrically connected cardiomyocytes in a very nice monolayer. And finally, we're very happy to report that the Cartox machine has been validated and used in other laboratories. So our friends at Fujifilm Cellular Dynamics have tested the Cartox machine over the last 3 months, and they were nice enough to share some of the data that they collected here with us. You can see in the bottom the timeline of their experiments, where they thawed cardiomyocytes, transitioned them to maintenance media, and then did assays. The recordings that you see on the right are from day 8 cardiomyocytes following thaw. And they tested the effects of pentamidine on cardiotoxicity. And this was done at different doses. And they used the Cartox machine to quantify the triangulation of the cardiac action potential. Any drug that increases the triangulation of the cardiac action potential is predicted to be more arrhythmogenic than compounds that don't increase the triangulation. And you can see in the right column, original recordings and analysis of action potentials in the presence or absence of pentamidine. And you can see the low with no compound, very nice regular rhythm of contraction. And with increasing dose of pentamidine, you can see that there's increased duration of the action potential, also slowing of the beat rate, and at the highest dose, even the occurrence of early after depolarizations and very high magnitude of action potential duration prolongation at the highest dose. So I'm going to leave it there, and I'll hand it over to my colleague, Monteiro Da Rocha, who will go into more details about the Cartox machine and the types of analysis that can be done using the Cartox optical mapping system.
All right, thank you, Todd. All right, so can you all see my screen? Hello?
Yes, sir.
Yes.
Yes, we can.
Yes. All right, so I'm really glad to be here talking about the Stambiosis approach to cardiotoxicity testing, which involves, as we've been covering throughout this series of seminars, like what kind of cardiomyocytes you should use for your tests and what happens when they go on Matrix Plus. And now we're going to focus on Cartox and the Cartox analysis software. And this will result in data that is used for cardiotoxicity prediction. So I think that we should all recognize that optical mapping for detection of action potentials, it's not something new. So the first work published was in 1985. So it's a technique that has been available for 36 years. But I would also like to put this in perspective to you, because this is very important. And the way that I'm going to do that, I'm going to trace a parallel between the Model T truck, right, that allowed farmers throughout the US to cover greater distance and carry their goods to markets and sell everything. So that was a big revolution. It was a big change in technology that was accessible and made everything easier for everyone, right? The thing is that there are, at that time, it was a great advancement. But if you compare a Model T truck with like a modern truck from the same company, I gotta say that if you're not a, you know, like vintage things, you will probably want to do your work using a newer model that has features that make everything easier. So I kind of feel the same way, and it's kind of the same perspective when we compare the optical mapping system that we've used to use to the Cartox machine. And again, I'm going to stress it out because if you haven't done it a lot, when you switch from an older system to this system, it's a huge difference. So although the two trucks work the same way, like the and have the same principles, the same thing happens to the optical mapping and the Cartox machine. The principles for optical mapping are the same. So we're gonna use a dye that can be, in our case, we like to use fluovolt, which is a membrane potential dye. So every time that there is a change in voltage, this dye will fluoresce. And also we like Calbright 520 AM for monitoring change in intracellular calcium. So the way it works, as if we're familiar with fluorescence, is that there is an excitation wavelength. In this case, it is blue. And every time that there is a change in fluorescence or intracellular calcium release, there's an emission of light in the green spectrum. And this can be captured by the optical mapping system. Just as a reference and to kind of give you the general idea on how it works, you have your plate with your cells, and it was loaded with either fluovolt or Calbright or Fluo4AM, if you prefer Fluo4AM. And it goes onto a temperature controller, and it gets illuminated in our case, it's trans-illuminated with blue light. And this blue light excites the cells when they're contracting or calcium has been released into the cytoplasm. And the emission of green light is captured by a CCD camera and in a high rate. And this data is stored into a computer for analysis of the action potential and the calcium transient duration. So this is, to us here at the University of Michigan, this is our Model T, right? So this is how an optical mapping system in an academic setting looks like. So I'm gonna give you a closer look to that, because there are a lot of things that if someone bumps a wire or, there are things that can go wrong because it's not a fully encased, and although we are used to it, it's intimidating for a lot of people when they start working with that kind of system. So we've been working, and as Todd mentioned, we had a SBIR grant to develop a new system for optical mapping. And this new system, there's a prototype here that you can see, has the LED illuminating the plate and the camera right above it, capturing the green light emission. So we were able to work with the STEMbiosis team to bring all those components together and make into a box. There are no exposed wires. It controls the temperature and has this nice drawer here that you open, put your plate in, as Todd showed in his slides, and you close your machine, and you're able to start recording your action potentials or the changes in calcium. The preparation of the samples is fairly simple. For fluovolt, you use 1 microliter of fluovolt per ml of HBSS and 10 microliters of power load per ml of HBSS. In general, the fluovolt kit comes with a background suppressor, but you don't need to use that. Actually, we've never used that because the signal is strong enough that you don't need to suppress the background, because the noise is despisable in this situation. So you replace your media with the diluted fluid volt. So for a 9.6 well plate, we use 100 microliters per well, and incubate that for 37 minutes, at 37 degrees for 30 minutes. And after the incubation period is done, you replace your dye with 37-degree HPSS or your drug treatment, and you incubate for how long you want. I mean, I'm talking about here about 9.6 well plates, but you can do pretty much any multi-well plate into the Cartox machine, right? So you replace, and then you're able to record. And for Calbright, it's pretty similar, although you still need to solubilize your dye in DMSO. The fluid volt comes already solubilized, and I like to dilute it in a way that I'm going to do a 1:1000 dilution. Then you dilute your Calbright in HBSS to a final concentration of 5 micromolar. We figured that this is enough to give you a pretty strong signal. They say that in the manufacturers of Calbright 520 AM say that you can go up to 10 micromolar, but 5 micromolar is enough, and it will not interfere with the pacemaker activity of your cells. So you replace your cell culture medium with Calbright, also 100 microliter per well of a 9.6 well plate, incubate for 30 minutes at 37 degrees, and then you replace your dye with HPSS for baseline recordings, or you can already add your drug treatment for the period that is more convenient to you. So this is just an idea on how it works. So here we have the 9.6 wells, and there are some ROIs here, some regions of interest, and this is what you collect, and this is unfiltered data. This is as it comes from from the machine without any filtration. But after you collect this data, there's a lot of processing and a lot of things that you can do to make the signals clearer. So the way that we do that is that StemBiosis developed a software that we call the Cartox analysis software to automate all this analysis. And again, this is one of those moments in which I feel like I just, came out of a Ford a Model T into a more modern truck, because if you were used to do optical mapping analysis with other types of software, you know that there were like sometimes multiple steps, and it was very time consuming. And for you to have an idea, a project that, or an experiment that would take me 1.5 days to 2 days to do, nowadays I can do it in 2 to 3 hours using this software, because it automates several steps of the process, and it also gives some really nice figured outputs. So once you collect your data, you will use this Cartox analysis software to analyze your data. And there are like 6 basic steps. So you need first to open or tile your movies into the software. Then you select your assay. You identify your wells. You select ROIs. If you want to select ROIs, it's not necessary, but sometimes it is important to do so. And after that, you select the analysis that you want to do, or the figures you want to generate. And on the last step, you just export your data as an Excel file for analysis. So this is how it looks like. And so when you open the software, you're going to be on this import filter tab, and you can ask to browse, which would allow you to open a single movie or tile multiple movies. So the Cartox equipment, it will take 12 movies from a 9.6 well plate, And once you ask them to be tiled, they will be tiled, and they look like this, but don't worry, because this is unprocessed movie, and it actually looks much better. Another thing that I want to point out is that in these specific examples, here we're not showing mono layers, but it's going to be clear in some other slides. We actually use a 96 well plate of micro tissues. So we've been working here in Todd's lab in making 96 well plates for production of high throughput engineered heart tissues. And you can see some of those right here, right? So you tile your movies, and then if you need, you can update your frame rate or the pixel size if you're gonna calculate conduction velocity, which is an important feature of this software. Then after you're done with this, you can come here to the well map wizard, and with one click, you're gonna activate the wizard, and you just click on the center of your corner wells, and it will automatically identify the wells. The other thing that is important, and it's just for organization, is to actually select which assay you're going to be using. And it's mostly because it will create a header that either says APD or CATD. And if you need to revisit a few years later or months later, what kind of dye and what kind of measurement was that, you already have that stored in your files, right? So after you're done with this, you click process save, and you're going to be able to go to your next window, right? And this is the window where you can select the ROIs, and there are many ways to do this. You can auto detect the ROIs, or you can add ROIs manually. So in this specific case, I did it manually because I wanted to have some rectangles over the micro tissues that you can see here, here, and here. And after you get your ROIs, you're ready to go to the analysis tab. In the analysis tab, you're going to have two groups of analysis, right? Here on top, you can have different chains you can select different percents of calcium transient duration. It can be 90, 80. Actually, here it is set up 10 by 10, but you can also enter it manually. Let's say you want to have a CATD 95%, you can enter it manually. You want a CATD 5%, you can enter manually. So it's pretty flexible. And some of the outputs that you're going to get are the frequency of contraction, the standard deviation of the frequency, calcium transient duration, triangulation. Then you have basal fluorescence, change in fluorescence, and then the amplitude, the time to peak, upstroke slope, calcium transient duration, and median conduction velocity of propagation of the intracellular calcium transient, or voltage if it is an APD that you're working with. The other things that you see here is that you can generate heat maps, and these heat maps can be for an activation heat map. So you're going to have a color-coded activation for each pixel that is in your ROI. You can do that for conduction velocity, for APD, and repolarization time. And if you click combine heat maps, it's going to combine every single ROI in a single heat map. Down here, what you see is the actual trace of the intracellular calcium transients, right? And here we calculated the calcium transient 80, and you can see where it's the 80 here with this black square. You can also select a single beat, and you get data from the individual that individual beat. And here you can generate other kinds of graphs and make an average trace or an average heat map that instead of being like a pixel, it will color code the whole well with the average of the calcium transient duration or whatever parameter you're looking into there. So this is one example. This is a time space plot of the microtissues. And here you have a microtissue that wasn't treated and a microtissue that actually received a treatment with isoproterenol. One thing that I didn't point out is that in this way, we could see that there is also an increase in fluorescence. So which means that there is more intracellular calcium being released into these cells after they're treated with isoproterenol. And there is also a chronotropic effect that the cells are, this microtissues started beating much faster. And you can also observe that with a time space plot. So I selected two areas, two ROIs, and then they were plotted here. And I generated a time space plot in which you have time here. And this is the spatial information on the X axis. So every time that you see one of these light green lines, it is an activation that happened. This is an example on how you can use the average trace. These are kind of stretched out, but I think that Todd showed some data that was generated by FCDI that shows like very well aligned average trace. And this is another tool that is like very interesting, is that you can make heat maps for the average of a single well. So here we have some optical mapping that was done with FluoVolt and Calbright and using different drugs. Here's in this quadrant here, we've used ranolazine that we know that blocks sodium and potassium channel. Here it's domperidone that is a dopamine antagonist that has hERG channel blocking effects. Nitrindapine is a calcium channel blocker, and vendetinib is a tyrosine kinase inhibitor used for cancer treatments that also has calcium and sodium and potassium blocking capabilities. This is a heat map that shows the frequency and how the frequency gets affected by the treatment. So here you can see that there is a little bit of slowing with increasing doses of ranolazine. Oh, and these are, by the way, these are the dose recommended by CEPA on their validation studies. So you can see that here we had a little bit of decrease in the bit rate as well as with domperidone, while nitrindapine, which is a calcium channel blocker, induced quiescence in the two higher dose. Another other important parameter is the APD80 that is automatically calculated for all your wells when you're using this tool. So ranolazine, it prolonged the calcium the APD80, and domperidone also prolonged the calcium transient duration. Nitrindapine, you can see that there was, it looked like there was a shortening, but then the cells stopped beating. And the calcium transient duration here was prolonged with vendetinib. And you can see, you know, it makes it very easy for visualization of your data. As Todd had mentioned, APD triangulation is an important parameter because it indicates if a given drug is likely to cause arrhythmias. So with ranolazine, we have a significant increase in the APD triangulation that can be seen here in this left top corner, right? So this is the lower dose and this is the highest dose. And you can see that it starts to get lighter, which means that it is being prolonged. With domperidone, we see a similar effect because it has, but in calcium, you can see that there is a prolongation in the recovery of calcium into the SR. Nitrendapine, again, those were quiescent, so we can see what happened in the highest dose. And with vandetanib, we don't see much of a change in the calcium transient duration triangulation. Here are some other ways that you can export your data, or you can show your data. You can copy individual trace, and this is a action potential trace as a baseline, and you can see the notch in the action potential, very well marked, and observe the changes that are induced by E4031. So you can get all these traces and combine them into an average trace, and they will look like this. So in purple, you see the average trace of a control atrial cardiomyocyte. And this is the same atrial cardiomyocyte, but treated with E4031 that induced this prolongation on APD80. And also change the morphology. Another thing that I didn't comment yet is that if you see here, you can calculate the upstroke slope of each of these curves. And that is a surrogate for sodium channel activation that is very useful. If you use, for example, dose response curve of meloxicam, you can see that there is a decrease in the upstroke slope that is correspondent to the increase of dose of meloxicam. Here again, we have ranolazine showing that the higher dose of ranolazine induce a prolongation of the APD. And here we have domperidone, and this is calcium mapping that also prolongs the calcium transient duration. Again, those are average trace that you can put together all your trace and compare what the doses are doing and which are the doses that are prolonging more of your action potential duration. And this is the domperidone calcium mapping with the dose response, in which you also see that there is an increase in the calcium transient duration in the highest dose. So these are just different ways to see your data after you use the Cartox machine. Well, and then you will probably want to do your stats, so you can use this other tab here. We're working actively now to have two settings, one for express use and the other one for more advanced users or that want to get more information about specific parts of the data. But you can group your wells. You don't need to, but you can do that if it makes easier for your analysis. And you can export your files as CSV, TSV, or Excel files. I recommend using the Excel files because if you do CSV, it generates a single file for each parameter. So you're going to have one file for your frequencies. You're going to have one file for the triangulation, one file for it. So if you use an Excel, you export this in as Excel, you have one file with different tabs that contain all your data, right? As I mentioned, you can come here and group your wells, and this will make it easier for you to do your statistical analysis after you're done. I think that this is basically how it works, and I gotta say that as a user, this like really improves the quality of data that I've been generating, the figures, and it also significantly reduced the amount of time for analysis of this data. And it also makes it much easier because all the equations and the calculations that you need to do, they're already in the software and can be changed for different levels of APD or CATD, which is like very handy. I think that's all that I had to present about the Cartox software. And I know that Travis wants to talk a little bit more to you about this improvement to the field.
Excellent. Thank you so much, Todd and Andre. It's a pleasure as always. So there's just a few things that I want to mention, and then I will begin going through the questions that are in the chat. And so feel free right now, if you had questions on the presentation, to go ahead and type those questions into the chat while I'm speaking. And when I get to the end, I'll begin reading those questions for Todd and Andre and myself to answer those. And so the first thing that I think bears mentioning is I always like to tell people that the heat map color schemes are highly customizable, so you can change what those are. I'm personally very colorblind, and so whenever Todd and Andre show those slides, they mean very little to me while they're in that color scheme. So those can be customized however you need them. I also want to note that while today we were really focused on presenting this technology that was developed by Cartox and STEMbiosis in collaboration, largely through the SBIR grant proposal that Cartox got awarded. I want to note that we understand that not everyone is going to want to analyze their cells this way, or at least not exclusively this way. And so we're happy to make whatever plates people want, and even collaborate on developing best practices. And so we are constantly adding new formats to our inventory that we carry, but also we're happy to make custom coatings when people need specific plate types for desired assays. And so some of the ones that we're routinely doing for customers now are things like seahorse plates, or putting these onto putting our matrix onto cover slips for people that are doing patch clamp assays. We've also had a number of customers requesting our substrate be put onto microelectrode array plates. The one caveat that I need to mention is that when we're making our coating for plates that we're not routinely using, we can stand by the fact that we know how to seed the cells and get a mature phenotype, but we may not be equipped to give advice on troubleshooting the assay if it's not one that we're routinely doing. But again, we're open to collaborations to develop best practices for different assay types that aren't in our current wheelhouse. I also want to note that the software and hardware that was presented today, the Cartox device and the Cartox analysis software, will be available for sale. So it's not on our website currently, but it will be in the future. And if you're interested in a quote, in the meantime, you can reach out to us. You can either email myself or Iris. There's iris.flores@stembiosis.com. And I am Travis, and travis.block@stembiosis.com. And we're happy to discuss this more with you, set up a call to talk about it one-on-one, and get you a quote on these solutions. And with that, I just want to remind everyone that this seminar, as well as our previous seminars from this series, are available on our website. And you can always reach out for more information, and these seminars will also be sent to everyone that signed up. And so with that, I'm going to open it up to questions. We have a few already in the chat. So the first one is Todd and Andre. I think this one's going to be best for you. So how does movement affect the readings? And so specifically, I think when we're talking about those micro tissues, does the cell movement, does the contractility change the relationship between the tissue and the pixels, and does that affect the readings?
Yeah, I can take this one. So Carlos, thank you for your question. It's like really a really interesting question and important. So I think that we need to think about the scale of things here. So in general, motion artifacts are reported when you have, for example, whole hearts, that when a whole heart contracts, there's a rotation of the heart and it's a huge amplitude of movement. When we're doing the micro tissues here, at least with the scale in which we're collecting the optical data, we were not able to see any change that suggest that we had motion artifacts for the size of the microtissues that we're working with. So these microtissues, they are about 2 millimeters long and 0.5 on width. So they're not that big, and their displacement, although you can use a microscope to measure the displacement, I'm not sure if we can see the displacement very well, at least at this point with the Cartox machine. But let me show it to you here again. Let's share my screen. So, right, these are, can you see my screen?
Yes.
Yeah, so you see how there's like no other chains than just the calcium going up and down, and these are from the microtissues. In general, we don't see a nice baseline when you have like motion artifact or you have like double peaks. And at least with this type of microtissues that we're using, we were not able to see alterations in tracings that suggest that we're having a huge deal of motion artifact. But again, I think it's a measure, it's a function of scale. If we were taking images that were much closer or using, for example, a 1x or a 4x objective, that would be a case to work on trying to remove the motion artifact. But at this distance, at this working distance, we could not see it.
Thank you, Andre. The next question that we had in the chat was, does nifedipine at low concentrations increase the beating rate of iPS-C cardiomyocytes?
I can take that question. So regarding calcium channel blockers, what we've found is nifedipine will shorten the action potential duration and speed the spontaneous beat rate. So definitely the effect that you would expect to see from a calcium channel blocker is certainly observed when we do optical mapping.
Excellent. Thanks, Todd. You know, one other thing that I wanted to add is, you know, Andre talked quite a bit about how this new tool that we've developed is making it much easier to get these measurements. But it's one thing coming from a lab that routinely does cardiac electrophysiology. But as their collaborator, I am not an expert on cardiomyocyte biology or any sort of cellular electrophysiology. And I can tell you that the software is simple enough that I'm able to take optical mapping readings and do the analysis in the software. And so the fact that I'm able to do it is a testimony to how intuitive the software is and how much it's simplified the workflow for analyzing these types of data sets.
Yeah, we had a lot of summer students that were able in the 10-week period that they stayed here to master it and generate data using the software. We've actually been like revisiting a lot of data that we've generated and was analyzed with Scroll. That was the proprietary software that we've used here at the Center for Arrhythmia Research. And I've been redoing a lot of analysis because data looks clearer and we can generate better figures. So I'm as someone that uses it very frequently, I'm really happy with the result.
Thank you, Andre. With that, we'll stay on the line for a couple more minutes, and then if there's no further questions, we'll go ahead and end the recording. But again, if you have any additional questions, feel free to type them into the chat and we will answer those.
We can also address any questions that you might have. I know there are multiple attendees on here that have actually had the ability to purchase some of the plates, the CELLvo Matrix Plus plates in different formats. If you have any questions about those, we're happy to address them at this time as well. And then if you can't think of the question or you think of it later, feel free to email us. You see my email right underneath my image, my tile there. So you can definitely email us at any point if you have questions that you think of after the presentation. But we will send a copy of this recording to you. I think I see another question in the chat. Travis, do you want to go ahead and address that one?
Sure. The question is, can you do repetitive measurements in a single well in long-term experiments? So can we do serial measurements using the Cartox device?
Yeah, that's a good question, especially for the chronic studies that are coming up for validation through SIPA. Yes, we can do imaging repeated measurements using the Cartox device. The Cartox machine is environmentally controlled, so it's kept at 37 degrees, and we do the recordings usually using HBSS, and the lid doesn't need to be taken off in order to observe those changes in fluorescence.
Do you have any a timeline? Did we already talk about that, Travis and Todd, about when to expect to have these instruments available?
So I don't have when it's going to be on our website, but if we received an order today, we're looking at a lead time of approximately 4 to 6 months. We're hoping that as supply chain issues get better, that lead time would be more like 12 weeks. But right now, what we're looking at is about 4 to 6 months.
Okay.
Yeah. So Iris, if you've mentioned before about like people ordering the plates from the Matrix Plus plates, so I would just like remind people that yes, you will be able to see your cells getting rod shaped in a regular plastic plate, but if you want to do immunofluorescence and fluorescency and confocal imaging, please make sure that you're getting the appropriate plates, because the matrix will be there. It will do what it needs to do to the cardiomyocytes. They're going to mature, and you're going to see the effects that we've reported here. But if you're trying to do confocal on a plastic bottom plate that the plastic's like very thick, you're not going to be able to go up to the magnification that you want. So please, if you're going to order plates and your intention is to run confocal experiments, make sure that you're getting plates that are optically correct for your application. And I know that STEMBiosis already have them on shelf.
Yeah,
That's correct.
just from, yeah, go ahead, Travis.
Oh,
I was just gonna give them a little reference point. I'm sorry, Travis, we both, that little time delay. So as Andre was mentioning, for reference, we do have CELLvo Matrix Plus available on our standard plates. So 6-well, 96, T75, and T150. That's what is meant by plastin bottom plates. But we also do EBD coated plates and Grainer microclear plates, and these both have a different bottom that's definitely better for confocal or fluorescent staining. So those are available on our website. You can see those as our stock items. And then, as Travis mentioned previously, we do have a custom service where if we don't have the if we don't have a plate with our coding on it on our website, this is a custom service. So you can just discuss with us what plate you'd like to see, and we can see if it's feasible, and then we can also get you a quote on and a timeline of when you can expect to receive it.
Yeah,
Go ahead, Travis, I'm
even
sorry.
seahorse plates can be coated with Matrix Plus. It's important to say, because we saw important effects on the mitochondrial activity in the paper that we reported, Matrix Plus, and people that are interested in exploring how mature cells work and how is their respiration, they can also get seahorse plates. We're actually getting some soon to run some experiments.
Soon. So seahorse, we've done seahorse, we've done auroras, some MEAs. We've also done different variations of the Grainer plates. So if it's if it's possible, we'll definitely do it. And Travis's team looks at feasibility from the start, just to see if this is a good plate to coat, and then we go from there. Travis, did you have any additional comments?
No, I think you summed it up nicely. If there's no additional questions, I think we can go ahead and end the recording. And I'd just like to thank everyone for joining us once again. We've really enjoyed putting together the seminar series.
Thank you all. Have a great rest of the week.
Yep, you too.
Thank you.
Thank you.
Yeah, bye.
Bye.
Thank you.